I'm going to talk about three different types of polymorphisms: SNPs, Indels, and CNVs. So, a SNP stands for single nucleotide polymorphism, and what this means is that a single base pair in your DNA has mutated from one base pair to another. In this case, we have a GA SNP. Because you have pairs of each of your chromosomes, you can be homozygous or heterozygous for a SNP. In this example, I'm showing you double-stranded DNA grouped by a green box. There are two green boxes in each example, and you should think about these as being a pair of chromosomes. In this first example, this person is homozygous for G at this SNP. The second is heterozygous. It contains both a G and an A SNP. And the third is homozygous for A. In this example, I'm showing you both sides of the double helix, but in future examples, I will only show you the coding strand. And now I'm going to talk about effects of SNPs. The first effect you might have is no effect. This can occur if a SNP occurs in a non-coding region of the DNA or in an intron, as seen in this figure. Another way to have no effect is if the SNP is in the third base pair of a codon. So in this example, you can see any possible SNP at this location would still result in a glycine. This is called the third base pair wobble, because regardless of the SNP, it'll code for the same amino acid. A mutation which does not cause an effect is called a synonymous mutation. A SNP could also cause a missense mutation. As you can see from the image, the A has mutated to a T, causing glutamic acid to mutate to valine. These amino acids are completely different. It's important to keep in mind that if an amino acid in the mutant is similar to the amino acid in the wild type, they might function similarly enough to not affect the end function of the protein. Unfortunately, that is not the case here, and this mutation causes sickle cell anemia. It's amazing to think something as simple as a single base pair mutation in the sixth amino acid in this protein can cause such a devastating disease. As a side note, being heterozygous for sickle cell confers resistance to malaria, and it is because of this heterozygous advantage that this mutation still exists in the population. The final effect of a SNP is a nonsense mutation. This means that the SNP results in a premature stop codon, resulting in a truncated protein. As you might expect, this is particularly bad if the SNP occurs toward the beginning of the protein, as more amino acids will be missing. An indel is an insertion or deletion. This occurs when one or more bases is missing or added, and is most problematic if 1 or 2, or really any number that isn't a multiple of 3 nucleotides is added, because it results in a frameshift mutation. Notice that the two sequences of DNA are exactly the same. However, when a single A is inserted into the bottom strand, all amino acids after this point will mutate. This is particularly devastating when it occurs at the beginning of a protein, since all amino acids after that point will likely be different. The most famous indel, simply because we found it here in what used to be the Laboratory of Genomic Diversity, is CCR5 delta 32, a 32 base pair deletion in the CCR5 gene.